Okay, so in the last video, we talked a little bit about different kinds of inheritance patterns that might be of interest to you or of importance to you as you're putting together your project on the inherited conditions. Okay, this video, I hope it's coming to you in time, but this video is intended to be a little bit of a guide, maybe a template for you to use as you're putting together your projects and presentations. By now, I'm hoping that you've already gotten some of the feedback from me with the personal research outline, the assignment that you guys completed. I should have given you some feedback on that relatively quickly. What I want to do is to give you some general information, maybe a few what to-dos and what not to-dos when it comes to your project. Okay. Now, one thing that I want to make it clear is that there's a tendency of some students to think, well, this is just like what I did back in the 9th grade when I took biology. I did a report on cystic fibrosis, right? And so I talked about this, I talked about that, and yeah, I included some of the inheritance there as well, but that was just a small part of it. What I really spent a lot of time talking about how it's diagnosed and how bad it is to have cystic fibrosis and how some people might be treated, how long they're going to live. Yeah, I mean, there are lots of things that are completely interesting about the conditions that, quite frankly, we're not terribly focusing on right now, right? This class is about the biology underlying these conditions, and that's what I want you to focus on, in particular the molecular biology, right? So it's not a report on condition X. So if you have already done a project on some inherited condition in your past, it's probably not going to work terribly well for you to recycle that report, okay? Just letting you know. Right? There are many parts to the project as indicated in the personal research outline assignment. And maybe this is going to be misrepresenting the actual thing that we're getting after here, because I actually do tell you that I want you to describe things like, well, what does the disease look like, right? How does it present in people that have the condition? What is the history of how it was first discovered and diagnosed? And what kinds of people are most affected by the condition? Is it young people, old people, members of particular age classes, or ethnic groups, or social castes? There's a lot of information that I think that really is important for you to include in your project. I just don't think that any of these should be the focus. Okay, so it's like we're looking at these things that you did for your personal research outline. Yes, include, okay, but it shouldn't be the main important thing that you're focusing on as you're emphasizing. The majority of your presentation should be about the actual molecular mechanism. Okay? And so this inheritance mechanism, this is what you want to spend all of your efforts on, because you have to get this first. If you don't do a good job with molecular mechanism and inheritance, you're not going to get a very good score on the presentation. It might be a very clever and elegant presentation. People might enjoy it a lot because you're talking about things that are of human interest, but this is not an assignment that is generating human interest. It's an assignment that is after the learning of the biology underlying the condition, right? And so similarly, when it comes down to the diagnosis and prognosis and treatment, yes, things that are of great interest to nurses and future health professionals. Again, not a focus, although it's definitely worthwhile for you to mention them, to talk about them to some extent, because yeah, I think you need to make a complete picture of your presentation on the inheritance and mechanism of this particular condition. It would make complete sense for you to include all of this stuff and all of this stuff. But again, if you make that the main focus of your presentation and only spend a little bit of time talking about the mechanism, that's not going to achieve the objectives of this presentation. Okay? And so instead of thinking about this as a report on condition X, you might actually think about this as a report on gene X, which, if mutated, will cause a condition. Okay, and that condition is kind of like what carries the day, but what we really are interested in is not the condition itself, but the gene that causes it, and how does that gene, if mutated, cause the disease that we're kind of focusing on, right? Okay, and so this is a part that I want to spend time talking about with you here, and I'll give you some example, a little bit of a template with the example of color blindness. Okay, and so I'm giving you the example of color blindness as an illustration. So I've already asked you not to select color blindness as your own topic, because it wouldn't be fair for you to have this lecture there as what you can do for your project on color blindness, right? Okay, so yeah, here's what we're looking for. Okay, so what I mean by the mechanism means it has to be dealing with a gene and a gene product, right? And we have to put things into the normal physiology in the context of what happens normally in a person that does not have the mutation. They've got the gene is in the normal form that's there in the majority of the human population, and the gene is going to be producing a gene product, probably a protein, right? Because you know that genes are coding for proteins. And so if the gene is coding for a particular protein, the protein would be the gene product, right? That is normally there. And this protein might do something that's required. What is the role in normal physiology that is not there when you have the mutation, right? Because if you don't have this context, then it's really kind of impossible for you to talk about the genetic condition which is resulting when you have a mutation to this gene, right? Because presumably if you mutate this gene, if you knock that gene out, or you alter it in a way that it makes a different protein or a defective protein, that's going to trickle down and have an effect on physiology, which is going to result in the diseased state, right? or the altered condition. And that's the whole thing that I want you to be focusing on, right? What does a normal gene product of this gene do? How does it normally function in a person who doesn't have the condition? What happens as a result of having a mutation in gene X? How does that affect the gene product? And how does the altered gene product ultimately lead to the way that the disease or condition presents in real patients? Right? And above and beyond this, yeah, it's a good idea for you to dress it up. You can talk about diagnosis, prognosis, treatment. You can talk about other things like the demographics. If you find something else that you want to include, that would be totally great. Okay? And so allow me to kind of demonstrate the kinds of things in a very informal way. I mean, your presentation will be looking a whole lot better than what I'm about to do because I haven't had the time to make pretty PowerPoint slides with figures and tables that might be useful. In fact, in particular with color blindness, it would be really cool for me to include a picture of what things look like with a colorblind person as opposed to what things look like with normal color vision. Right? I mean, that's the sort of thing that you can totally do with your PowerPoint, and I'm not even going to try to do that. I think everybody here has got a pretty good idea about what I mean by color blindness, and so I'm going to go ahead and work with this. Right? So I'm going to be going through this part really quickly because you know that Color blindness is generally having a deficiency in the ability to distinguish between red and green. There are actually two forms of the most common X-linked color blindness, right? There is one called protanopia, and the other one is called deuteranopia. So there's the P form and the D form. We're going to be talking about one of them because I think that, the most that if you do a really good job on your presentation, I mean, if color blindness is caused by 3 different mutations, right? And it is, right? There are 3 different kinds of cones that can be affected. You've got the long wavelength cone, you've got the medium wavelength cone, you've got the short wavelength cone. Now, these are the 2 that are affected by the protanopia and deuteranopia. And there's a third one that we're not even going to talk about. If I only focus on this gene, that will be enough, okay? Because the whole process is required to track the disease from an altered version of the gene all the way through the actual presentation of the disease. That's what I'm asking you to do, right? Okay, so here we go. Let's take protanopia. There's a gene that's called opsin1, OPN1, and this is the long wavelength version of it. If you want to look up these other ones, not because I want you to do them, I want you to kind of steer clear of color blindness altogether, right? OPN1, M1, MW, medium wavelength, and I think it's OPN1, short wavelength. Let's say for the time being that these 3 genes are off limits for your presentation, right? So this is the gene that normally results in a gene product that produces the opsin pigment. Let me undo that. You spell pigment with an NT at the end, don't you, right? Okay, so opsin pigment, which is required to distinguish the long wavelengths. And at this point, I might be talking about how in the colors of the rainbow, we've got a spectrum ranging from red to orange to yellow to green to blue to indigo to violet, right? And so maybe you learned about the colors of the rainbow, Roy G. Biv, right? And so these are the long ones, these are the short ones over here. And so a person that has the normal opsin pigment is able to detect and distinguish these colors really well, right? So we're going to be looking at these colors and saying, yeah, I'm seeing those reds and oranges and yellows really nicely, right? And if you were to knock out, okay, you gotta change the colors. If you were to think, well, what would happen if you've got an opsin1 long wavelength gene that is defective? Then you're not going to be able to produce this opsin pigment, which means that you're not going to be able to see these colors, right? It's not that you're not going to be, you'll be able to see the light, but you're not going to be able to distinguish those particular wavelengths, right? You've got plenty of other photoreceptors to detect the presence of things and the outlines, the images, and what they look like. The one thing that you won't be able to process is what colors they are if they're reflecting light in the red, orange, yellow wavelengths, right? And that's in a nutshell kind of how you could kind of track what's going on from a gene. And its normal gene product, it allows us to see in these wavelengths. If you don't have that gene product, you're not producing the pigment and you're not seeing those wavelengths. Things are not going to appear red or orange or yellow to you, okay? Now, what could you do to dress this up? I mean, you could talk about diagnosis and prognosis and treatment. I would kind of like you, if possible, to talk about things that are more kind of biological interest, right? And so the fact that humans have three pigments, three cones, right? You've got the long wavelength, you've got the medium wavelength, you've got the short wavelength. That is actually different from most mammals, like dogs, cats. They have only two wavelengths, two cones. So they only have the long ones and the short ones, right? Now, yeah, the evolutionary story behind this is completely fascinating because as mammals, our history has been one of, at least if you go back, some 50 million years ago, most of our ancestors throughout the, more than 50 million years ago, if you go back to the Mesozoic, when mammals were kind of in the shadows of the dinosaurs, mammals were mostly nocturnal. And as nocturnal animals, you don't really get a chance to see colors very well. And so we lost cones. We basically lost the ability to distinguish colors because distinguishing colors was not terribly important to our life as nocturnal predators, right? Now, if you look at birds, which are basically dinosaurs, and I mean, birds are dinosaurs, which have had a pretty good representation in daytime living throughout. Birds and dinosaurs retain what is probably the ancestral state, which is to have four different cones, four different color receptors. Yeah, that's right. Birds' color vision is way better than ours. They're able to detect a broader spectrum and able to detect colors with greater precision compared to humans. Right? Humans do better than dogs and cats, and the reason for that was that in the lead up to some type of dichromat, a two-cone ancestor, when humans' ancestors became diurnal and started feeding heavily on things like fruit, where it was important to see colors. Okay? It was of great advantage for us to have better color vision. And what happened was that this long wavelength cone here in our ancestral state, the long wavelength cone, the short wavelength cone has stayed the same. The long wavelength cone split, and basically we got two copies of the long wavelength cone. And one of these copies evolved into a medium wavelength cone, which basically means that we've got not really a good covering of the spectrum. We've got kind of like a very long wavelength cone, which covers the reds and the oranges and the yellows, and a slightly shorter wavelength cone, which covers the oranges, the yellows, and the greens. And basically, if you don't have that, this is our medium wavelength receptor here. If you were to lose the medium wavelength cone, you get a slightly different version of color vision where you actually see a little bit of the reds, but you lose more of the greens, right? And so that's the deuteranopia form of color blindness. Not quite as common as protanopia, but definitely still relatively common. Okay. Other things that we've already talked about in past classes is how this is carried on the X chromosome. I would have to include that. Being on the X chromosome, it's going to be far more prevalent in males because males being X, Y, are going to have just one copy. If you have the mutated version of the X chromosome, you will be displaying color blindness, right? And if you're a female, however, you have the opportunity to have a regular, normal opsin-producing gene to fill the gap, to make up for the presence of the mutant gene, and therefore females are, even though you're carrying the gene for color blindness, you're actually color sighted because you've got the good copy on the other X chromosome, right? And so the only way for a female to actually have color blindness would be for them to be XC, XC. And this is going to happen far more rarely. And so that's why if you've got males at around a 1 in 20 likelihood of being colorblind, because one X chromosome out of 20 in the human species is the one that carries the color blindness gene. The prevalence in females would be the likelihood of getting a colorblind gene, which would be 1 in 20, times the likelihood of getting a colorblind gene in your other parent, which is going to be 1 in 20 again, which is going to give you 1 in 400. That's the likelihood of actually being colorblind as a female, like Jeff's mother-in-law. Okay. I hope I didn't put those calculations in the part of the screen where my picture shows up, but you got to hear it anyways. And so in other words, so you saw what I did. I mean, I want to spend most of the time talking about the way in which this gene is giving rise to the condition that I care about, that I'm focusing on, and then including everything else, like the way that it's inherited, the way that we actually ended up with these genes might be a good thing to add in the case of how we ended up with the normal genes is part of the story in the case of color blindness. Okay, and and this was especially important in the case of color blindness because there's not a whole lot of things that I would be able to tell you that you didn't already know about color blindness, right? Yeah, people that have color blindness are not able to see colors normally. Okay, big surprise there, right? No, so I think if you're finding yourself reporting something that everybody would know anyways, that might be a good thing to not include in your actual presentation, or maybe dress down, not spend a whole lot of time focusing on it, talking about it, and spend your time talking about things that would really inform your audience, keeping in mind that your audience is, at this point, a pretty sophisticated group of biology students. All right? Hope that helps.